Animal cell culture. One important approach to studying the cells of multicellular organisms is to grow isolated cells in culture, where they can be manipulated under controlled laboratory conditions. The use of uh, cultured cells has allowed studies of many aspects of mammalian cell biology, including experiments that have elucidated the mechanism of DNA replication, gene expression, protein synthesis and processing, and cell division. Moreover, the ability to grow animal cells in culture has allowed studies of signaling mechanisms that control cell growth and differentiation within the intact organisms. Although the process is technically far more difficult than the culture of bacteria or yeast, a wide variety of animal cells can be grown and manipulated in the culture. Cultures are initiated by the dispersion of a piece of tissue into a suspension of its component cells, which is then added to a culture dish containing nutrition media. Most animal cells types, such as fibroblasts and epithelial cells, attach to and grow on the plastic surface of dishes used for cell culture. Because they contain rapidly growing cells, embryos or tumors are frequently used as starting material. Embryo fibroblasts grow particularly well in culture and consequently are one of the most widely studied types of animal cells. Under appropriate conditions, however, many specialized cell types can also be grown in culture, allowing the differentiated properties to be studied in a controlled experimental environment. Embryonic stem ACs cells are a particularly notable example. These cells are established in a culture form early embryos and maintain their ability to differentiate into all types of the cells present in adult organisms. Consequently, embryonic stem cells have played an important role in studying development and differentiation, as well as offering the possibility of contributing to the treatment of human diseases by providing a source of tissue for transplantation therapies. The initial cell cultures established from a tissue are called primary cultures. The cells in a primary culture usually grow until they cover the culture dish surface. They can then be removed from the dish and replaced at a lower density to form secondary cultures. This process can be repeated many times, but most normal cells cannot be grown into culture indefinitely. For example, normal human fibroblasts can usually be cultured for 50 to 10 population doublings, after which they stop growing and die. In contrast, embryonic stem cells and cells derived from tumors frequently proliferate indefinitely in culture. They are referred to as immortal cell lines. In addition, a number of immortalized rodent cell lines have been isolated from cultures of normal fibroblasts. Instead of dying as more of their counterparts do, a few cells in these cultures continue proliferating indefinitely, forming cell lines like those derived from tumors. Such permanent cell lines have been particularly useful for many types of experiments because they provide a continuous and uniform source of cells that can be manipulated, cloned, and indefinitely propagated in the laboratory. The first human cell line to be established were HeLa cells, which were isolated from a cervical cancer in 1951 and have been used in thousands of laboratories studying many aspects of human cell biology. Viruses. Viruses are intercellular parasites that cannot replicate on their own. They reproduce by infecting host cells and usurping the cellular machinery to produce more virus particles. In their simplest form, viruses consist only of guanimic nucleic acid surrounded by a protein coat. Viruses are important in molecular and cellular biology because they provide simple systems that can be used to investigate the function of cells. Because virus replication depends on the metabolism of the infected cells, studies of viruses have revealed many fundamental aspects of cell biology. The rapid growth and small genome size of the viruses have made them especially important for studies of mammalian cells. Most animal viruses replicate and can be readily studied in cultured cells, where they take over the machinery of cell to produce new virus particles. The genomes of animal viruses are much smaller and simpler than those of cells, ranging from approximately 3,000 to 30,000 base pairs and often containing less than a dozen genes. Animal viruses are thus far more manageable than their host cells, making it comparatively easy to follow virus replication and undertake genetic analysis. Examples in which animal viruses have provided critically important models for investigation of mammalian cells include studies of DNA replication, transcription, RNA processing, and protein transport and secretion. It's also noteworthy that infection by some animal viruses can convert normal cells into cancer cells. Studies of such cancer-causing viruses, first described by Peyton Rose in 1911, not only have provided the basis of our current understanding of cancer at the level of cell and molecular biology, but also have led to the elucidation of many of the molecular mechanisms that control animal cell growth and differentiation. Tools of cell biology, microscopy, and subcellular fractionation. As in all experimental sciences, research in cell biology depends on the laboratory methods that can be used to study cell structure and function. Many important advances in understanding cells have directly followed the development of new methods that have opened novel avenues of investigation. An appreciation of the experimental tools available to the cell biologist is thus critical to understanding both the current status and future direction of this rapidly moving area of science. The two fundamental methods of studying the structure of cells, microscopy and subcellular fractionation, are described in the sections that follow. Other experimental approaches, including the methods of molecular biology, genomics, and proteomics, will be discussed in later chapters. Light microscopy. Because most cells are too small to be seen by the naked eye, the study of cells has depended heavily on the use of microscopes. Indeed, the very discovery of cells arose from the development of the microscope. Robert Hooke coined the term cell following his observation of a piece of cork with a simple light microscope in 1665. Using a microscope that magnified objects up to about 300 times their actual size, Anton van Leeuwenhoek in the 1670s was able to observe a variety of different types of cells, including sperm, red blood cells, and bacteria. The proposal of cell theory by Matthias Schleiden and Theodor Schwann in 1838 may be seen as the birth of contemporary cell biology. Microscopic studies of plant tissues by Schleiden and of animal tissues by Schwann led to the same conclusion. All organisms are composed of cells. Shortly thereafter, it was recognized that cells are not formed de novo but arise only from division of pre-existing cells. Thus, the cell achieved its current recognition as a fundamental unit of all living organisms because of observations made with the light microscope. The light microscope remains a basic tool of cell biologists, with technical improvements allowing the visualization of ever-increasing details of cell structure. Contemporary light microscopes are able to magnify objects up to 1,000 times, since most cells are between 1 and 100 nanometer in diameter. They can be observed by light microscopy, as can some of the larger subcellular organelles such as nuclei, chloroplasts, and mitochondria. However, the light microscope is not powerful enough to reveal fine details of cell structure, for which resolution the ability of a microscope to distinguish objects separated by small distances is even more important than magnification. Images can be magnified as much as desired, for example by projection into a larger screen, but such magnification doesn't increase the level of detail that can be observed. The diffraction of light limits the resolution of the light microscope to approximately 0.2 nanometer, two objects separated by less than this distance appear as a single image, rather than being distinguished from one another. The theoretical limitation of light microscopy is determined by two factors: the wavelength of visible light and the light gathering power of the microscope lens, numerical aperture (NA). According to the following equation, resolution is 0.61 wavelength divided by numerical aperture. Several different types of light microscopy are routinely used to study various aspects of cell structure. The simplest is bright field microscopy, in which light passes directly through the cell and the ability to distinguish different parts of cell depends on contrast resulting from the absor- ab- absorption of visible light by cell components. In many cases, cells are stained with the dyes that react with proteins or nucleic acid in order. Prior to staining, specimens are usually treated with fixatives such as alcohol, acetic acid, or formaldehyde to stabilize and preserve their structures. The examination of fixed and stained tissues by bright field microscopy is the standard approach for the analysis of tissue specimens in histology laboratories. Such staining procedures kill the cells, however, and therefore are not suitable for many experiments in which the observation of living cells is desired. Without staining, the direct passage of light doesn't provide sufficient contrast to distinguish many parts of cells, limiting the usefulness of bright field microscopy. However, optical variation of the light microscope can be used to enhance the contrast between light waves passing through regions of the cell with different densities. The two most common methods for visualizing living cells are phase contrast microscopy and differential interference contrast microscopy. Both kinds of microscopy use optical systems that convert variation in density or thickness between different parts of cell to differences in contrast that can be seen in the final image. In bright field microscopy, transparent structures such as the nucleus have little contrast because they absorb light poorly. However, light is slowed down as it passes through this structure so that its pace is altered compared with light that has passed through the surrounding cytoplasm. Phase contrast and differential interference contrast microscopy convert these differences in pace to different differences in contrast, thereby yielding clear images of live unstained cells.